Hi, hello everyone. In this video, we shall discuss about transpiration definition, types of transpiration by Dr. J. Ramudu, lecturer in Botany, VSR Government Degree and PG College, Mova, Krishna District. So, dear friends, I am going to discuss about the transpiration and the different types of transpiration and structure of stomatal apparatus, distribution of the stomata and stomatal frequency, types of stomata. Okay, my dear friends, so what is meant by transpiration? So let us see the definition of the transpiration. So transpiration means the loss of water from the aerial parts of the plant in the form of water vapor is called transpiration. So transpiration is loss of water from the aerial parts of the plant. So by the means of the aerial parts. So the aerial parts may be the stem or maybe the leaf or maybe the lenticels. So here the leaves are principal organs of transpiration. So most of the transpiration takes through the stomata. The stomata present on the leaves. Okay, this is about the transpiration. So coming to the next one is types of transpiration. So types of transpiration is, first one is stomatal transpiration, and the second one is cuticular transpiration, and third one is lenticular transpiration. Let us discuss one by one. The first one is stomatal transpiration. So the transpiration through the stomatal pore is known as stomatal transpiration. Generally the stomatal transpiration occurs in the plants 85 to 90% of the transpiration, that means the water loss by the stomatal transpiration. Second one is cuticular transpiration. So loss of water through the cuticle, but the amounts of loss of this water vapor very small amount and make up only about 5 to 10% of the total transpiration. So cuticular transpiration just 5 to 10%. And the third one is lenticular transpiration. So lenticular transpiration means that the transpiration through lenticels. So lenticels of the plants. So lenticular transpiration was about 0.1 to 0.2. So the total transpiration loss in this means just 0.1 to 0.2%. So coming to the next one is, so here the structure of stomatal apparatus. Here the chief organs of what loss of water vapor by the means of stomata. So now let us discuss the structure of stomatal apparatus. So what is the structure of the stomatal apparatus? Here we can see the diagram. Stomata was first discovered by Pfeffer in the name as stomata. So actually the stomata was given by the name by Malfigi. You know, stomata cover 1 to 2% of leaf area. So it is a very minute pore present in soft aerial parts of the plant. So you know, algae, fungi and submerged plants do not consist of stomata. So when we are observe the algae, whatever the algae is it green algae or whatever it is the algae, they could not find any stomata. And the second one is fungi. So in fungi we could not find any type of stomata. And also very important, submerged plants do not possess the stomata. Yeah, here the stomata are very minute pores of elliptical shape, consist of two specialized epidermal cells called guard cells. So the stomata made up of with particularly so elliptical shape of the cells that are those are guard cells. So the guard cells are kidney shape in dicotyledons, whereas in monocotyledons these cells are dumbbell shape. So you know the wall of the guard cell surrounding in the pore is thickened and inelastic elastic nature due to the rest of the walls are thin, elastic and semi-permeable. So the guard cells are the protective cells. Next one, when we observe the structure of the stomata, we can find out the guard cells as well as subsidiary cells. Here this is the dicotyledon stomata. In dicotyledon stomata, the guard cells are kidney shaped cells. So here these are guard cells, this is stomatal pore. Okay, right, coming to the point, when observe the stomata, we can find out the surrounding parts of this guard cells. We can find the subsidiary cell. So these are all subsidiary cells. When we find out these subsidiary cells, there are 2 to 4 are in number. Okay, so here we can find out the stomatal apparatus. So stomatal apparatus, that means stomatal aperture. So this is the structure of the stomata. So the subsidiary cell also consist of chloroplast. The subsidiary cells also consist of chloroplast. This is the just the structure of the stomata. So next one is, here the guard cell has the cytoplasmic lining, central vacuole. So the cytoplasm consists of a single nucleus and number of chloroplasts. So the chloroplasts of the guard cells are capable of the very poor photosynthesis because of the absence of Rubisco enzyme. Here when we observe this the subsidiary cells, here we can find that is the photosynthetic pigments, that is chloroplast. Even though the presence of the chloroplast, we could not find the Rubisco enzyme. That's why so the photosynthesis activity is very slow. Okay, next one is, guard cells are surrounded by modified by epidermal cells known as subsidiary cells. So here we can find out the subsidiary cells. Subsidiary cells or accessory cells which supports in the movement of guard cell. So here we can find out the subsidiary cells beside or above the that is the guard cells. So next one is, so the size and shape of the stomata, the guard cells vary from plant to plant when full open. So the stomatal pore measure 3 to 12 in width and 10 to 14 in length. So when we observe that width and length. So coming to the next one is the number of stomata. So the presence of the number of stomata on the leaf is known as stomatal frequency. So the number of stomata per unit area of a leaf is called as stomatal frequency. So the number of stomata in a definite area of leaf varies from plant to plant. So you know the xerophytes possess large number of stomata than the mesophytes. So here they can the evidences that there is the morphological as well as physiological evidences prove that the xerophytes consist of high number of stomata when compared to the mesophytes as well as the hydrophytes. Okay, coming to the number of stomata, that means square in centimeters, 1000 to 60,000 in different plant species. So the number of stomata per unit area of a leaf is called stomatal frequency. So in a unit area, how many number of the stomata present, that is called as stomatal frequency. So next one is distribution and types of stomata. So depending upon the distribution and arrangement of the stomata in the leaves, we can categorize it into 5 types. So of stomatal distribution have been recognized in the plant. So there are 5 types of that is the stomata. One is apple or mulberry type of the stomata. So we call as the hypostomatic stomata. So these stomata are found in distributed only on the lower surface of the leaves. So if the lower surface of the leaf, we can find this type of stomata. So this type of stomata is known as hypostomatic stomata or apple or mulberry type of the stomata. Second one is potato type of the stomata. The stomata are found distributed more on lower surface and less on its upper surface. So for example, potato, cabbage, bean, tomato, pea consist of more stomata on the lower surface and less stomata on its upper surface. So this type of stomata is known as a potato type stomata. So the third one is wart type. So wart type also called as amphistomatic type of the stomata. In this stomata, they are found in distributed equal upon two surfaces. So the two surfaces upper or lower. So adaxial as well as abaxial. Example, maize, oats, grass. So when we find the maize, oats and grasses, we can find equal amount of stomata distributed both upper as well as lower surface of the leaf. So the fourth one is water lily type of the stomata. So this type of stomata are found and distributed only on the upper surface of the leaf. So the stomata could find only upper surface of the leaf. For example, water lily, nymphea, many aquatic plants consist of only upper surface of the leaf could find the stomata. So this is water lily type stomata or epistomatic type of the stomata. So the next one is potomaceton or astomatic type. So stomata are altogether absent or if present they are vestigial. Example, potomaceton and submerged aquatics. We could not find any type of stomata on the leaves or the plants, or if there is a presence of the stomata, so those are vestigial. Those are vestigial. So these are the based on the distribution and arrangement of the stomata. The stomata categorized into the 5 types. So next one, Metcalfe and Chalk recognized 4 types of stomata on the basis of their structure. So on the basis of their structure, the stomata categorized into different types. The first one is Animosity Stomata. So in this stomata, accessory cells are absent. The guard cells are surrounded by the ordinary epidermal cells. So when we find out this, this is the Animosity type of the stomata. So the guard cells are surrounded by ordinary epidermal cell. So example, so for example, Ranunculaceae, Cucurbitaceae, Papaver and Malvaceae, we could find the accessory cells absent, only the guard cells are present. So this is Animosity Stomata. So Animosity Stomata present only the family members like Ranunculaceae, Cucurbitaceae, Papaver and Malvaceae. The next one is anisocytic type of the stomata. So in this type of stomata, guard cells are surrounded by 3 accessory cells. So when we find out this diagram, second diagram of above the photograph, we could find the anisocytic type of the stomata. So anisocytic of the stomata found in the family Brassicaceae. So of these 2 are larger, where one is smaller in size. So here there are 3 surrounded cells. The stomata, these stomata guard cells are surrounded by the 3 accessory cells. The accessory cells, 2 are large cells and 1 is small cell. This type of stomata found in Brassicaceae. Next one is diacytic type of the stomata. So this is the third one. So in this type of the stomata, the guard cells are surrounded by 2 accessory cells. So their common walls are at right angle to the walls of the guard cells. So families of Caryophyllaceae or Acanthaceae, these are the best example of diacytic type of the stomata. Next one is fourth one, parasitic type of the stomata. So in this stomata, the guard cells are also surrounded by the 2 accessory cells, but their common walls are parallel to guard cells. So example is family Rubiaceae and as well as Fabaceae family. And so these are Animosity, anisocytic, diacytic and parasitic stomata. So that's the second categories of classification of the stomata. Next one is on the basis of the development of the stomata, Pant 1965. According to Pant 1965, there are three types of the stomata. One is mesogynous type of the stomata, perigynous type of the stomata, mesoperigynous type of the stomata. So what is meant by mesogynous type of the stomata? So in this type of the stomata, the guard cells as well as subsidiary cells or accessory cells both are developed from only one mother cell. So example, Rubiaceae and as well as Brassicaceae families or this type of stomatal development. This is mesogynous type of the stomatal development. So in this type of stomata, guard cells as well as subsidiary cells or accessory cells both are developed from only one mother cell. So next one is perigynous type of the stomata. In this type of stomata, the guard cells are formed from mother cell while the subsidiary cells from nearby mother cell. So here that is the different type of the mother cells. So example is Cucurbitaceae. So Cucurbitaceae is the best example for the perigynous type of the stomatal development. Next, the third one is mesoperigynous type of the stomatal development. In this type of development of the stomata, the guard cells and one subsidiary cells is formed from mother cell while the other subsidiary cells developed independently. Example, Ranunculaceae, Caryophyllaceae families. Okay, so thank you very much.